Yes, thank you. So in my postdoc, I worked on the very, very beginning of development. Oh, thanks. Yeah, this is better. So in early embryos, like a zebrafish embryo that you see in this video, the cytoplasm needs to be robustly organized into cells. And to achieve this, large microtubule structures that are called asters, they fill the space in the embryo, reading its geometry and dividing the content into compartments. But actually what is very interesting is that if we look at these stages, we see that this compartmentalization is already achieved before the cell membrane ingresses. So this raised the question for me, like, of how can the cytoplasm be robustly organized and partitioned without physical barriers consisting of a cell membrane. So to study this process, in my postdoc, I used frog cytoplasmic extract. And by looking at this compartmentalization, I discovered an instability that is driven by microtubule aster growth. So you can see on the video on the left that microtubule asters, they grow and they start invading each other, and they drive the coarsening of the cytoplasm and the relocation of many nuclei into these very large multinucleated assemblies. So this is a big problem for the embryo. However, because this instability has a time scale that is related to the growth of the aster, if the cell cycle, cell division is fast enough, it can actually make the system temporarily stable and allow for robust development. Something that I saw is that also that the asters that actually are involved in this instability are very large and they have a lot of branches. And this is the case for frog and fish embryos. However, when we look at other species like insects, for example, like Drosophila, asters are much smaller and they are characterized by less branches. And actually we can show that this makes them stable. So an interesting thing of actually looking at the physical parameters of microtubule growth and plotting them in this phase diagram is that we see that microtubule turnover, like other parameters, are quite conserved across the species, but actually it's the number of branches that is tuned across these different organisms. And if you come to my poster, I will tell you later how changing the number of branches of microtubules actually can drive different patterning strategies in these embryos. So finally, I would like to thank HFSP especially for their cross-disciplinary fellowship, because I'm a physicist by training, and the grant opened up to me the world of cell and developmental biology. And it also funded this project that will be the base for what I will do in my future lab, that I will start at the Hubrecht Institute next year. My lab will work mostly on cytoplasmic organization and the transition and emergence of multicellular system from a physical point of view. Thank you.